Hi, my name is Julie Caruso, and I'm a science teacher at Star Spencer Mid-High. And I'm going to walk you through the instructions and topic, what role does DNA have in how traits are passed through generations. All right, so this week we're going to be covering how mutations or mistakes in transcribing or translating DNA to RNA affect organisms, proteins, things like that. So you're going to want to read through all of this carefully. Make sure that you're starting with a hypothesis. Always put it in writing so you can go back and check to see what you've learned or how your opinions have changed. So do that first. And then you're going to work your way through this first set here, which is asking you to translate the DNA into the mRNA using this codon wheel. Now, if you're having trouble seeing this, I recommend that you Google it. Keep in mind, though, when you Google, a lot of the codon wheels that are going to come up here are going to have a different layout than the one that is in the packet. So you're going to see GU or AG. The one in the packet has ACUG, this order here. It really doesn't make a difference, but if you don't want to confuse yourself compared to what you're seeing on your packet, make sure you find a codon or a codon wheel that has ACUG at the top. But again, this isn't made up material here. This is the actual codon wheel. So it doesn't matter which letters are on top. The groupings are going to be the same surrounding it, so feel free to use whichever one you like. Going down, make sure you're filling in the chart here. Looking at it carefully, you want to look at the analyzing the amino acid sequence, protein structures, things like that. And they're asking you to try to determine why it is that you think that if it's miscoded, that it looks differently. And you'll see the differences down here, being circular in shape here, or that sickle or crescent moon kind of shape that you'll see with sickle cell anemia. So looking at the conclusion aspect of this, you're going to compare it to your hypothesis, and you're going to write a CER here. Make sure you're using academic language, make sure you're using the proper vocabulary, and writing in complete sentences. So activity for the next day is reading into gene expression, and we have an online version here, or you were given the printed copy, and it is a Khan Academy article, as you can see here. And keep in mind when you're reading through that the words in blue are things that will link you to another article for more information. And if you look at the menu over here on the side, you're right here in what is a string of information about molecular structure of RNA, DNA, transcription, replication, all that stuff. So if you're confused on how we got to this point, read the articles below it. And if you're interested in learning more afterwards, follow through with the articles underneath. There's an extra website for additional information. And then the next day, we're asking you to draw some models. You have two choices below here, drawing a model of DNA and RNA or drawing a model of transcription and translation. One are the two types of molecular sequences here, DNA and RNA. What do those strands actually look like? And the other one is the action of transcription or translation. What does that look like? So they are two different things there. And the next day, for Demonstrate Your Learning, you have several options that you can choose from here. Write a story about what could happen if humans began to edit DNA using CRISPR. And if you're not entirely sure what CRISPR is, there's an article down here in your extensions. And you could choose draw a model that shows how DNA is converted into protein. So you're looking at the transcription and translation process. Make a comic strip that shows the story about the discovery of DNA. And there's some people down here you'd need to do some research on to make that a good comic strip. Explain the concept of the central dogma of molecular biology. And if you're not familiar with that one, I highly recommend Googling it. As you can see here, if you type in central dogma microbiology, it will come up here with this brief description of it. And then down here, it'll, if you click on some of the people also ask questions down here, it'll give you a little more information about why they call that central dogma and, of course, what central dogma is. You could also write a tweet that explains the structure of DNA and why it's essential for life, or, and this is kind of along the same lines, create a social media post regarding sickle cell anemia and its causes. And down here, it's giving you specifically how sickle cell anemia is caused by a DNA mutation that changes an amino acid from glutamic acid to valine. So make sure you're paying attention to what the actual details of this particular assignment is. Reading the CRISPR article. If you come over here, it's a Newsella article about doctors using CRISPR to edit for blindness. CRISPR is a huge part of microbiology and what is new and upcoming in science. So this is some good stuff to know for those of you that are interested in learning more about microbiology or gene editing in your future. So have fun with the extensions. There's a music video here. If you have any questions at all, reach out to your biology teacher, and we hope that you enjoy this week's lessons for biology. Take care.